Today we will talk about plant and animal cells. See, we know cell is a basic functional unit of any organism. You know, very tiny organisms are single cell organisms or single cellular organisms. We all, whether they are plants, animals, or whatever you see around you, we all are multicellular organisms. That means there are billions and trillions of cells which work in a specific coordinated manner. Each cell is a separate functional unit. Like in human body there are trillions of cells. So basically the number is so huge, but each cell, of course there are cells dedicated for some specific purpose, they are also there. There are a variety of types of cells, but in general all cells have some commonness or some similarities. Even plants and animal cells, there are two separate kingdoms that way, plant kingdom, animal kingdom. But the cells from both the kingdoms are also similar in many ways. And yes, there are some differences. So we all are multicellular or eukaryotic creatures with so many cells. So we will try to understand what is inside of every cell. Plant cells are normally rectangular in shape. So like a brick with some corners polished, but that appears to be a rectangle in form. Animal cells are more towards round or little irregular shape. Yes, furthermore, plant cells are bigger than animal cells. Size-wise, they are up to 100 microns, plant cells. And animal cells are up to 30 microns. So, animal cells are small, they are not exactly rectangular, they are oval, circle or some different shape. That is a fundamental thing. Now, we will go for the parts which are inside and which of course have boundaries and everything like that. So cell, particularly if I talk of plant cell, this is plant cell. What I have drawn here is plant cell. Has a thick cell wall. So cell wall is there in plant cell but absent in animal cell. I have written P and A for sake of just getting some clarity. The cell wall is made up of cellulose mainly. It is thick, protective membrane. Inside of cell wall, there is something called as plasma membrane or cell membrane. It is there even here. That is the way cell interacts with its surrounding. So exchange of anything happens through this semi-permeable membrane. That is called as plasma membrane. And inside it is filled with nothing but cytoplasm. It is like a liquid, thicker than water, but it is liquid that is filling both the cells. So again, this is common. So plasma membrane is common, cytoplasm is common. And inside this there are several other small elements or organelles which work independently, and this becomes a functional unit. We consider each creature as a single unit, like a human being. But inside humans also there are so many separate units working together, like cells. So it's very interesting to know what is inside. Both the cells will have a nucleus. But the nucleus here is little shifted towards one wall. And here of course it is almost in the center. So this holds the genetic material of that organism. What is that genetic material? That means from generation to generation some traits they transcend or they continue. That happens because of DNA which is inside of your nucleus. Every cell has that DNA inside. So that is passed on to next generation. So every cell basically has that kind of parentage for next generation to come in. That is your nucleus. Both of them they have it. Then there are some ribosomes or ribosomes, which are circular and they are scattered around at different locations. I just draw them here. And here also there are some ribosomes. Basically, they are small spherical organelles which process proteins. So ribosomes are for processing of proteins or production of proteins. Okay. So this is about ribosomes. One thing I should draw before I draw other organelles, that is vacuole. Vacuole is nothing but some space which is like a ball, you could say, which creates some different membrane bound space. This has a large vacuole of the kind, and this may have small vacuoles, one or two, but they are small vacuoles. I use two different color paints. So if I call this a vacuole and these are ribosomes, this vacuole occupies sometimes even up to 90% of volume of plant cell. That's the reason this nucleus is shifted on one side. Vacuole occupies maximum middle space. This is important. It stores, of course, some kind of starch type of liquids also, but also gives that sturdiness, toughness to the plant cell. Here vacuole, either one or more, they are very small. Okay, so that is about vacuole. Then chloroplasts. Chloroplasts are, gifted only to plant cells. They are oval in shape, having some kind of folded. There are multiple, so I am drawing 2, 3. They are responsible for making energy for every cell. And we know the plants are called as autotrophs, which generate their own energy by trapping sunlight. So that happens inside these chloroplasts or plastids. Plastids are also, there's a term used for chloroplast and something more than that as well, but we can call them chloroplasts here. So they trap sun's energy, that is converted to sugars, or that is what we call as energy. So it is chloroplast. Around this nucleus, there are endoplasmic reticulums. They are some random structures surrounding nucleus, like a stack of some, you can say zigzag kind of pattern. Some of them may have these dots on them. They are nothing but same ribosomes which are coming here. I'll draw them with a red color if that needs some clarity. So they are ribosomes. They are called a rough reticulum, and those who do not have a soft reticulum. This again has production of proteins happening here, and they are also used for some kind of detoxification of, if something goes wrong with the cell, that is removed by this smooth reticulums. So that's the purpose of reticulums. And rough reticulums, which are seen with dots, these dots are nothing but ribosomes. They produce proteins, and these proteins are then passed on to one more apparatus that is called as Golgi apparatus. Here also we have these reticulums around nucleus, and there are some Golgi apparatus. So these proteins which are produced are transferred to Golgi apparatus in both the cases. So the reticulums and Golgi apparatus, these two have discussed. Okay. Chloroplasts we have discussed. Mitochondria. Mitochondria are mainly seen in animal cells. Maybe one mitochondria could be seen in plant cell as well. But here mitochondria, they are usually oval shaped, or they are irregular, and they have these kinds of folds inside. And mitochondria is power bank or powerhouse of the cell. Here that formation of or burning of sugars actually happens. That is oxidation of sugars with help of oxygen that is coming from your breathing comes here, it processes or it oxidizes that sugar and produces high energy rich molecules called as ATPs, and they are kept there. So basically that is like a powerhouse, storing power for this cell and of course in turn for you. So each cell has these powerhouses called as mitochondria. Then this is about something which is common. I will go for something which is exclusive in animals. Peroxisomes or lysosomes. These are some organelles which are unique for animal cells. Peroxisomes particularly, they do one specific thing. From any substance which is like, for example, if poison or anything, if it has entered the cell, it takes out hydrogen from it, forms hydrogen peroxide inside in the cell, and that helps. That those toxins are then of course removed, or their functionality is killed by peroxisomes by taking out hydrogen from it and forming hydrogen peroxide. Lysosomes, very interesting. They eat whatever unwanted comes inside the cell. For example, some bacteria enters or some virus enters. That is engulfed by these lysosomes. So they are practically protective mechanism for cell. Also, if some organelle becomes dead or it breaks, then that dead content or broken content is also eaten away by lysosomes, and then of course given out by this cell into the bloodstream or some other streams which are moving inside your body. But each cell has these lysosomes to take away that unwanted part, bacteria or something. Animal cell also has centrioles. Centrioles are nothing but cylindrical structures which are holding a special role in reproduction of your cell. Cell when splits, then this plays important role in splitting of DNA, and then formation of new cell is also controlled by centrioles. So coming back to this, vacuole is big in plant cell, small in big cell. Chloroplast present, absent. Size is big, small. Plant, animal. I am comparing this. Lysosomes, it is absent, it is present here. Reserve food. See, plants have reserve food in the form of starch. We say starchy food, like potato and so on. They are starchy foods. But in animals, it is reserved in glycogen, in the form of glycogen. So some differences are definitely there. We have discussed about the cell wall and so on. So I have not written everything out here. You would now understand these all lysosomes, peroxisomes, centriole. Centriole is a crystalline structure of this kind. So that is also there. All these things make up a cell. And see, a cell of size between 10 to 100 microns for plants and 10 to 30 microns for animals looks very small. It's a microscopic object. We know that. We cannot see a cell by naked eyes. But even a single cell holds such a variety of things. I have not written at least a handful of other things which are there, but I have not written them. So understand that cell is a separate unit which works independently. It has everything. Like it takes things, and there are even cilia which are used to give out the material outside. This is also a semi-permeable membrane giving out the things outside, and so on. So every cell is filled with so many organelles, parts inside. We will now try to make model of these two things. So now is the activity time for you to make cell model for a plant cell and animal cell at the same time. Remember one more thing. Whenever we place some small parts on that platform of plant or animal cells, be sure that you have understood it. It is not just a foam piece you are just making some kind of zigzag structure and placing it. You have understood working of those all internal organelles of a cell. Then only it will make sense, and you can explain cell structure using a big cell in your hand and explain the role of every single part of that cell. All the best. Let us do the activity now. We will start making a model of plant cell to begin with. Plant cell is a rectangular form. However, we need to cut the corners. So I'll do one thing, I'll just draw pencil lines to mark these corners. And then using a cutter and scale, we'll just remove these four corners. You should do this carefully because you have to handle a cutter. So take a small portion, not big. So keep just using this cutter in one direction. So one part is cut. Likewise, you have to do at all four places. You have to take approximately same size, same corner triangle each time. And the last one now. So the main difference between plant cell and animal cell is its cell wall. So once basic structure is done, we will place a cell wall around this structure. So this is a cell wall. I'll do one thing, I'll just place it around it in such a way that its edge is little up. Okay, so we'll start placing it and finally we'll cut the unwanted part. So I need to use a lot of pins. Just put them into the foam at different locations. So this will represent cell wall. You know, plant cell has cell wall that is made up of cellulose. Make sure that this is tightly placed around this foam, and that will look nice if its outline is brought little up over this foam. Overall, for this activity you may need about 50 to 60 pins. For complete making up of this plant cell, you may need 30, 40 or even 50, 60 pins. Okay, now let us avoid this overlap. So I may mark it using a pencil and we'll cut this extra portion. So I can use a pair of scissors, it will be easy to cut. So I have cut it and we'll just join them together to make it a continuous cell wall structure. So this is almost done. So this placement of cell wall. Okay. Then we'll go for, so you know, there is a major size or volume occupied by a vacuole. For vacuole we have this foam structure. So we can make vacuole of some large size like this, and it may not be a regular size. You can give any shape to this. Again, start placing it using a pin, using pins. You can press it if you wish to make a odd shape of this, not exactly uniform oval, but something odd shape of it you can make. So you need to place few more pins. Okay, so we have placed vacuole. Now for placement of nucleus, we need to use a pearl headed pin along with a large sized bead together. Usually this nucleus comes in one end of cell. See, while pressing this long pin, it is advised that you press it in a tilted way, so its other end does not come out of this foam. Okay, so this nucleus is also placed. Now we need to show a reticulum. For reticulum, we will be using this foam structure. If this much is sufficient, we'll cut it here first. We may use two different reticulums. Just fold it like this and place it to represent reticulum, which is usually near nucleus, so I am placing it here. The reticulum is smooth and rough. There are two types of reticulum, smooth reticulum and rough reticulum. We need to make it rough by using crushed marbles onto it. We may place one more small piece of reticulum here. There are two or more such strands of reticulum in every cell, and part of it is smooth, part is with ribosomes on top of it, that is rough reticulum. So that is done. Now we will apply some Fevicol on top of some part of this reticulum, not everywhere, and place this crushed marble powder on top of it. That will represent roughness of this. So this is rough reticulum part. Okay. Now next comes is Golgi Apparatus. We'll place Golgi Apparatus somewhere here. See, the placement of these all organelles could be anywhere. There is nothing very specific. But normally Golgi Apparatus comes close to reticulum, so I am placing it close by. So this much of Golgi Apparatus, again it is folded. So the Golgi Apparatus, it comes like a tubular structure. I am making a tubular structure using this tube. This is rubber tubing. I am placing it here. Now, putting pins through this rubber tubing may become a little tough for young kids, but not very difficult. Now, functions of each organelle, each part of this cell are given in the notes and discussed even in theory of it. But you can refer to the notes in case if you find that you have forgotten the theoretical part which we have discussed in videos. So this Golgi apparatus done here. Now we know that plant cell has chloroplasts. So chloroplast is the specialty of plant cells. They are kind of oval in shape like this. We'll place maybe 2 chloroplasts somewhere. One and maybe one more. This is a clay I am using, plasticine clay. So make a flat oval shape of this clay and you can place anywhere. Remember that you are not placing it inside vacuole. These are all outside of vacuole. This vacuole, center one. So don't use that place. Now out of these two, on both of them, rather one after another, I will to place a thread making a zigzag shape. I'll make 2 equal parts of this thread and we'll use them one on each of them. So just by pressing it against this, you can form a zigzag shape. Just to show how exactly this plastids or chloroplasts look like in reality. So if there is excess of thread, we may cut it. I'll try it with other chloroplasts as well. Same structure. You know, it's a representation of different things. So naturally you have to show how exactly it appears when plant cell is seen under microscope. Now this again I'll try to place in a zigzag way. We'll insert some pins through this so it will remain in place. Same for this. Okay, so this is done. Then in plant cell there may be 1 or sometimes little more than 1, maximum 2 number of mitochondria. Mitochondria will represent using this foam. Mitochondria is again irregular in shape, usually oval. So I am just making an oval out of this rectangle like this. It is not very regular, so any shape would do. Okay, so this is what I have created. Let us place it somewhere here. There is a mitochondria. In case if you wish that you can just reduce the size of this vacuole because this is occupying too much of space. I'll just reduce its size a little bit. This also has that kind of zigzag pattern. I'll achieve this using a rubber band. We'll place a pin here likewise. Let us see if we can stretch a rubber band over these surfaces. One more. And then we'll press it down. Let's see if this remains in place. Okay, fair enough. Otherwise you can just twist the rubber band and lock it. Now this tubing, I am cutting its 2-3 small pieces, almost of same size, and we'll place them inside of this vacuole, because there are some crystals which are inside vacuole. I'm just placing them using again same pins. There are some crystal forms inside vacuole, so they are represented by these tubes. Okay, now finally we may place some 3-4 number of these pearl headed beads again outside, again tilted. These are representing ribosomes. So they are scattered everywhere. They are needed for protein formations in a cell, and will place a very small piece of this tube, because you know that in a cell food is stored in the form of starch. So you can say this is starch. So the plant cell stores food in form of starch. So this is a plant cell almost ready. Maybe we can place peroxisomes, this white colored one to represent peroxisome. So these are ribosomes, this is red or purple colored, peroxisome, starch. This is your chloroplasts or plastids. This with rubber band, once again we have to place it, or you can pin it over this rubber band, that's also fine. And your cell is about to get ready. So once you finish with this, you would rather put labels to this, so that you will easily be able to explain function of each organelle using this model. So basically this is the model making we have done. Okay. Now everything done. The cell, plant cell, it just complete. Big vacuole. This is your nucleus, rough reticulum, smooth reticulum. All these ribosomes, these colored beads, you can see. This small is not to represent ribosomes, but it is to represent peroxisome. Then this is your mitochondria, Golgi apparatus or Golgi bodies, and these two, which are important part of your plant cell, is your plastid or chloroplast, and the entire content of this cell, cytoplasm, is shown green, because that is the way plant cell is characterized by. So cytoplasm, vacuole, and everything shown properly in this plant cell. We will prepare animal cell using this foam as the main base or main cell. You know, cell is filled with cytoplasm. Now this represents that fluid which is called as cytoplasm. Here we should know that animal cell is smaller than plant cell. It is about 10 to 30 microns in size, whereas plant cell is 10 to 100. Now besides, one more thing, this should be a kind of roundish or irregular in shape. It is never rectangular like plant cell. So as a first step, we need to cut these corners this way. So I'll do one thing, I'll mark them with pencil or any pen. So all four should be marked. Even a scratch is fine, that you should know where to take the cut, and you should give a little round shape while taking the cut, so that looks nice. First, remove these four corners carefully. And you need not worry about the exact cuts, like, because it may not be round, it may be irregular. So holding this, I am just cutting it. I may change this a little bit. So start cutting it. Apply blade only in one direction. And once you cut this completely, you may use cutter to soften the corners so they become round. You should take care that you don't cut your finger or table surface. So I am going little slow, so I don't want to cut the table surface. So one side is now done. Okay, then I just use the same cutter to give it a soft corner. Okay, in the same way I'll cut other three corners and make it look like a round. Now this is the shape of animal cell. As I told you, it is never exact circle. It is little irregular. So we made it that way. It does not have cell wall. So there is a membrane, but that cannot be shown. We will begin with placement of a nucleus. We'll be using this bead, a big size bead, and a pearl headed pin to place this. Place it somewhere in the center. So this is your nucleus. Around nucleus there is reticulum. Reticulum is a structure which is to be formed using this kind of foam. See, it is surrounding this. So we may take it like this. So make it something to look like a curvature made using this foam. Place them using pins. Place these pieces using pins. So I am placing pins. One is placed here, one is placed here. So this first curve is done. So you need several pins for making structure of cell, whether it is plant cell or animal cell. These all are called as organelles inside a plant or animal cell. Right now, of course, it's animal cell that we are doing. So insert it at several places. You may use few more pieces and place them around. Using pins again, same way. So this is reticulum. Now reticulum is used for synthesis of proteins and of course few more functions. It is of two types, one is smooth and one is rough. We'll just convert them to smooth and rough once it is done. Okay, so this reticulum, shape is not certain, so you can give any shape to this. Now we will use this crushed marble powder to create a rough surface of reticulum. I am taking this little bit of crushed marble powder. So I am placing this marble pieces or marble powder on some part of this reticulum to convert it to rough reticulums, maybe here as well. So these are basically small ribosomes on these reticulums. You know, ribosomes are the places where again proteins are synthesized. I am representing these ribosomes using pearl-headed pins. Remember that these pearl-headed pins are little longer, so you should insert them in a slanted way so they do not come out from the other side. So these are pearl-headed ribosomes we have placed. So good enough, we have placed 4 of them. Anywhere, wherever you feel like. Now then we have to place Golgi bodies. They come in continuation of this actually, so I'll just use this tube to form Golgi apparatus or Golgi bodies. I am cutting it somewhere. Let us form this Golgi apparatus here. Again you need to fix it using pins. So the protein comes here from rough reticulum. So Golgi apparatus is done. Now we need to show other organelles. The important ones, of course, they are these. I am making a oval shape to show mitochondria. They are more in number. We'll show 2 of them. So again there is irregular shape of it, so we'll use a red foam for representing mitochondria. It has some zigzag kind of structure inside. We'll use thread to form those zigzags or that structure typically. So these mitochondria, single one is called as Mitochondrion. So we'll use a thread piece. I'll just place it in a zigzag way on one of the 2 mitochondria. So this I'm placing it in a zigzag way. Additionally, we'll put pins on top of it. So let us use pins. Now we have created 2 mitochondria. These zigzag pattern created using thread and pins. Here also it created using thread and pins. So 2 mitochondria. Now animal cell also has small vacuoles, 1 or more. So we'll create a small circular vacuole using a black colored foam. We'll take a small piece of this foam and convert it into a round shape representing vacuole somewhere. So this is fixed now, that vacuole part is done. Now what remains are 2-3 small things. There is something called as lysosomes. We'll place this using another pin in this place. So these are large size lysosomes. You can place 1 or 2 such molecules or organelles. These are lysosomes. And one small additional organelle should also be represented, that is called as peroxisome. I'm using this white colored thermocol bead for it. So this is so far done. Now it also has some microtubules. So I'm just making small pieces of this tube. 2 or 3 of them are enough. And place them using again pins. It could be at any place. So microtubules are there in cell. I have taken few wooden sticks fixed in a rubber band this way. They will, these 2 sticks together will represent centriole. That is one more exclusive organelle in animal cells. So you should press this down as much as you can. And this is centriole. This helps in division of animal cell. So this plays a role. And now to summarize all the organelles inside animal cell. It does not have cell wall. It has a nucleus almost in the center with this nucleotide. Then around this we have reticulum. Reticulum is either soft or rough. This is rough and soft reticulum. Then these are nothing but ribosomes on reticulum. These red beads are these ribosomes inside. This is zigzag pattern of Golgi apparatus. Then this centriole, as I told you just now. These 2 red colored with zigzag pattern of thread are mitochondria. This vacuole, which is small in animal cell. These 2 red beads, they are representing lysosomes. White bead is representing peroxisome. Then we have these microtubules. That's all about. And this is already told to you. So these are different organelles inside animal cell. Now you can explain each one of them and with these actually model parts and explain their function, their placement and so on.